Our genetic information, handed down for generations, holds the code to who we are. The intricate process of a small cluster of nucleic acids can shape the course of our lives is fascinating in and of itself, but particularly through the lens of a 1 in 20 million genetic condition, Hutchinson-Gilford Progeria syndrome. Hutchinson-Gilford Progeria syndrome is an autosomal dominant genetic condition with symptoms of premature aging, among others, that are present at birth but become more prominent in early childhood. Unfortunately, individuals with this condition often succumb to it in their teen years, although new treatment options are improving this prognosis. Let's start by defining autosomal dominant. Dominant refers to how this condition is passed down. In general, offspring receive only one copy of each gene from each parent, called an allele. Progeria being dominant means that an individual only needs one allele to be affected and must therefore have an affected parent. Since progeria interferes with fertility and sufferers rarely reach sexual maturity, this scenario is almost impossible, unless in cases of mosaicism, so progeria is typically caused by a random genetic mutation. Autosomal means that the mutation that causes progeria is not on a sex chromosome. In people with progeria, their element A gene codes for progerin instead of pre-lamin A. Pre-lamin A eventually matures into lamin A, which is part of the nuclear lamina, a scaffolding that gives the nucleus its round shape and likely plays a role in regulating gene expression. Progerin, on the other hand, does not serve this purpose, and its accumulation causes DNA damage as well as cell death, leading to the symptoms associated with aging. Like all other genes, element A is read by RNA polymerase, which uses it to transcribe a strand of pre-mRNA. However, not all of this information is made up of useful exons. For this purpose, the genetic material has certain sequences, like punctuation, signaling when to splice off introns or junk DNA. The issue occurs on the 11th exon of the element A gene, where one point mutation messes up this punctuation, thus prompting the sequence to be spliced prematurely. Fast forward to the two RNAs being translated into proteins. Note that progerin is missing 50 amino acids when compared to pre-lamin A. The proteins are supposed to undergo 4 processes meant to transform them into mature lamin A. First, farnesylation. Second, proteolysis. Third, methylation, and the fourth step is where things begin to differ. The protein from earlier is supposed to cleave in the middle of the missing 50 amino acids in progerin, but is unable to do so. Instead of becoming a mature lamin A, progerin remains in its mutated form, contributing to the progression of progeria. The FDA approval of lonafarnib in November of 2020 is enabling individuals with progeria to lead healthier lives. Lonafarnib, as a farnesyltransferase inhibitor, limits the buildup of progerin within the nucleus, alleviating certain progeria symptoms. While this is not a perfect cure, as no lamin A is being produced and does not remedy the damage already caused by progerin, it is a step towards a brighter future for HGPS patients worldwide.